and I think that it's wonderful what they do to make science accessible to other people. So I am delighted to be here today to speak to you about my most recent paper, which has been the focus of my postdoctoral work as part of the Christophe Benoit and Diane Mathis lab at Harvard Medical School. We published it in January this year. And it's for any of you that want to access it. It should be available on the website of the CBDM lab. And so this project really started because I've been passionate about the gut for a long time. I've always found it fascinating how I had an immunology degree, and the first thing we're taught is how all of the immune cells have evolved to fight pathogens, right? And then we get to gut immunology, and suddenly turns out that we carry millions of microbes in our gut, and our immune cells and these microbes happily coexist and have evolved to need one another. And I always found that fascinating. And what's even more fascinating is that that environment doesn't exist in isolation, but as has been clear in the last, I'd say 10 to 20 years, is really what happens in the gut doesn't stay in the gut. So there's these axes, if you will, that have started coming up of, connections between the gut and other tissues, the most popular of which I'd say is the gut-brain axis. I'd say most people now are familiar with the idea that there's a connection between the gut and the brain, but, it's also with other tissues like the lung or the liver that we're starting to see more and more reports coming out linking these tissues. But the majority of the work at the time, especially about 5 years ago when I started doing this work, was focused around metabolites and more, we know there's a connection, but we don't really have the mechanism. We don't know how any of this is happening. And so, what we set out to do is, what about immune cells themselves? We know that immune cells have the ability to move. They've always had this ability to, respond to infection, travel to where infection is. They are mobile by nature. And there was some evidence published by the lab, I'd say about 10 years ago, where we knew that gut cells had some degree of mobility. So we wanted to really explore what was the extent of that phenotype, right? And so the first aim that we had when I started this project was to really understand how is it that the gut communicates with the periphery when it comes to immune cell migration. What's the extent of this relationship? And to do that, we mostly utilize the Caeta mouse model. This mouse model is great for this purpose. It was developed by Japanese scientists in 2008. And the Caeta mouse expresses a fluorescent protein called Caeta, which is green. And if you shine a light of the right wavelength on it, you can photoconvert the protein from green to red. So you can kind of see it here in the whole mouse itself. You can see the gradient from photoconversion all the way from green to red. And then down here, you can see that we're able to utilize this model and using an endoscope photoconvert the colon. So you can see this is like a section that we imaged using confocal microscopy, where you can see that on a regular Caeta mouse, the colon is green throughout. But if we use an endoscope to photoconvert, you can see that we can photoconvert cells throughout the whole lamina, all the way from the epithelium and through the lamina propria. And so the first thing that we did was study immune cell dynamics in the colon. So if we take these mice and we photoconvert them, and then we use flow cytometry to image green versus red cells over time across up to 3 days, what happens? And what we found is that It didn't matter which immune population we looked at, whether it was B cells or gamma delta T cells or mononuclear phagocytes, CD8s, T conventionals, Tregs. We always found that the cells that we photoconverted would drop over the course of those 3 days. And in some populations, more so than others, like for instance, B cells, by the day 3, we could barely detect any photoconverted B cells. And so this told us that, okay, there's huge turnover of these cells in the gut. And that could be for various reasons. Maybe they're proliferating. If cells proliferate enough, they'll lose the Kaida red signal, or maybe they're dying. But since we were interested in migration, what we found was actually, if we looked in the periphery, a lot of the cells that were missing from the gut could be accounted for by migration. So you can see it, it's very clearly a distinct population, where here you're looking at what we've used to generate a lot of the data that I'm going to show you. It's essentially just looking at green versus red expression by flow cytometry. In this case, I'm showing you B cells versus T cells. And this is in the spleen 24 hours later. So this is in a mouse where we photoconverted only the colon. So there should only be red cells in the colon. And we know for a fact, because we've run all the different controls, that if you look at time 0 in this mouse across different tissues, the only red cells are in the colon. So if 24 hours later, we're looking at the spleen and we're seeing all of these red cells, we know that they must have come from the colon. And so this to us demonstrates extensive migration out of the colon. And it's not just the spleen. So for a lot of our data, we use spleen as a reference, since most of our immune cells we assume are going to pass through the spleen at some point, and we can get great numbers to do further assays. But once we started looking at other tissues like inguinal lymph nodes, which are draining the skin, bone marrow, skin, liver, lung, kidney, blood, small intestine, we could find migratory cells throughout, with 2 exceptions. So in the thymus of adult 6 to 8 week B6 mice, we couldn't really see any migratory cells. And in the skin, surprisingly, because initially I thought, oh, it's another microbiota carrying tissue, wouldn't it be interesting if the 2 are communicating? But surprisingly, it was actually one of the tissues that seems to be not really being reached by gut cells. But anything else that we checked, we could find cells that were in the colon 24 hours before. And if we look at the identity of those migratory cells across the tissue, so down here, I'm showing you data that comes from flow cytometry and has been plotted onto a t-SNE across the different tissues. And I think what becomes obvious very quickly is B cells are a pretty dominant population, which was surprising and unexpected, considering I was part of a T-cell lab. This wasn't the expected result, but it's very clear that the B cells are a dominant population in the spleen, but the T cells are also there, and also some myeloid populations to a lesser extent. And then as we looked through the tissues, there were some similar themes, but also subtle differences. Like we could see a lot more T cells migrating into the lymph nodes. And then some of the more non-lymphoid tissues, like the lungs or the liver, you could start to see some of the other more rare populations being a bit more dominant. Like we could find a lot more disease, for instance, in the liver compared to the spleen. And so we thought this was, of course, very interesting. I think this opens up a lot of possibility, because at this point, we're still looking in healthy mouse that, as far as we know, are not perturbed in any way. So this is just a snapshot, but we're assuming that this is something that's happening, this exodus of cells that are reaching other tissues is happening constant in a sort of conveyor belt, if you will. And one thing that we did wanted to account for, because when I first started talking about this and showing this data, there was some resistance in terms of, oh, well, how do you know that you're not just capturing, cells are moving. So maybe you're just photoconverting cells that are just passing through, and then of course they're going to move. But we found that as far as we're aware and as far as the data that we have in hand, we don't have anything to indicate that that is the case. We ran an experiment where we labeled circulating cells using a CD45 antibody via IV. And what we found is actually, so if you look at the top row here, via flow cytometry, we're looking at the three biggest populations, the mononuclear phagocytes, T cells, and B cells. And the percentage that actually comes from circulation, it's very low. We're looking at 1 to 2%, versus the amount of cells that we're actually photoconverting. It's huge difference. And then at the very bottom here, if we look at the photoconverted cells, which are then labeled from circulation, it's basically 1 to 2 cells. It's an extremely low percentage. So this, in our hands, can't explain what we see in the periphery. So we think that the cells that we pick up in the spleen and in the peripheral tissues are not just cells that were passing through the column, but we think that they were part of the tissue at the time of photoconversion. And so at this point, two other questions emerged. One is, how are these migratory populations different from their non-migratory counterparts, and do they carry any gut imprinting programs, or once they leave the gut, they forget they were in the gut in the first place and they just become circulatory cells? That was one question that we had. And the other main question that we wanted to answer was, all of this, like I said, has been done under steady state, but what happens when steady state is breached, if there is disease of some kind or infection? And first thing that we did was we wanted to profile the difference between migratory versus non-migratory cells. And to do that, we used a single cell experiment where we photoconverted the colon, as we have been doing, for unchecked 24 hours later, 48 hours later, and 72 hours later in the spleen. And we sorted green versus red cells in the spleen, pulled them all together, and did single cell sequencing. And what we found initially was we recover the populations that you would expect to see in the spleen. You've got your B cells, T cells, and a few of the other more rare populations. And if we look at the distribution between Candida red and Candida green across those cell types, you can see that it reproduces what we had seen through flow cytometry, where B cells are very dominant, but really we can find Candida red cells across all of the different populations. And if we started focusing on each of the populations, so obviously the B cells were the most dominant, so that's where we started. When we mapped the population of B cells, we found the populations that you would expect from the spleen. So you have your B follicular cells, your cycling transitional stage cells, some marginal zone cells, really mature IgM positive, B cells that have seen interferon. What was surprising was whenever we looked for the Candida red cells, as you can see here in this plot, what's depicting is in the area that's bright red, it's where the Candida red cells are dominant. And then the areas that are blue, it's where the green, or what we assume are spleen resident cells, are dominant. And first observation is that the migratory cells are not sort of separating into their own population. They seem to fall within the rest of the B cells in the spleen. And what was surprising as well is it seems that they most closely resemble B follicular cells, which are not what one would find generally to be a migratory population. So that was somewhat surprising. And then there were some populations that seemed to be unrelated, or at least not profiled within the migratory cells, like the most mature IgM high population. We couldn't really find a lot of migratory cells there. But even though they weren't really separating into their own separate population, we next wanted to ask how different are they from the ones in the spleen? And so when we did differential gene expression across the most dominant chita red clusters, what we found is that we actually could find a sort of gene signature that separates the chita red from the chita green cells. What you can see here is there were certain groups of genes that characterized the chita red cells, so the ones that came from the column, regardless of time of photoconversion. So whether they were photoconverted 1, 2, or 3 days before, there were certain groups of genes that seemed to be consistently expressed across all of them. There were some that appeared to be enriched only in the ones 24 hours later. And then there were some that appeared to be downregulated, as well as some that were downregulated throughout, regardless of time of photoconversion. Some of these modules are related to metabolic pathways. And then a lot of the ones that are associated with the migratory cells, especially early on, have to do with activation as well as migration. And so that's the B cells. Then the T cells, on the other hand, were actually pretty different. So whenever we profiled the T cells, we did find distinct, more gut-related populations. So you can see here, once we mapped in the UMAP the various T cell populations in the spleen, again, it's what you'd expect. So you have your T conventionals, your CD8s, your Tregs, then activated T cells, a group of RA gamma T cells, separate cluster of mostly CD160 positive T cells over here, and some NKTs. And if we look at the distribution of the cells that came from the gut versus the one that were, let's call them spleen resident. So the reds fell mostly around, you could find them in practically every cluster except the NKTs. There weren't really NKTs other than 2 cells that came from the gut. But a lot of the reds actually fell within the activated T, the RA gamma T cluster, and the CD160T cluster. And what became a bit curious to us is we were a bit puzzled initially by this CD160T cluster because it was composed of both gamma delta and alpha beta T cells, and it didn't have anything standing out to separate them as any particular effector cell. But once we started looking at the gene expression of that particular cluster a bit more closely, it seemed to resemble a lot of to the intraepithelial lymphocytes that we find in the gut, so IELs. We can't really say they're definitely IELs, but it is the cell type that they resemble the most, which was intriguing to us. And then after that sort of initial broad profiling, we started to see if we could get a bit more information around them. One thing we did was, since the cells came from the gut, what would happen if we put the mice on antibiotic? So I put the mice on antibiotic for 10 days. And the reason I did 10 days is we didn't want to do a full course of antibiotics that would significantly alter the composition of the immune cells in the gut. And so by doing 10 days, as far as we could tell, the composition of immune cells in the gut was still basically the same as in a wild type mouse, but the microbiota was already gone. And so at this sort of early stage of antibiotic microbiota depletion, if we looked at migrations of cells into the spleen or even the skin draining lymph nodes, we could already see a decrease in the migration of cells, especially the two most affected populations were the B cells and the gamma delta T cells, but some of the others were impacted as well. And as a final piece of data that I want to show you today is a bit of a teaser to that second part of the question of what happens if we start doing further perturbations, not just something as benign as antibiotics, but actually disease. And so we chose 3 different disease models. So one was the arthritis KBM model, which Christophe and Diane pioneered. Then we also chose a tumor model, which is the MC38 model, which we use subcutaneously. And then we also used the model of skin inflammation in the ear, which is the DTH model. And we followed similar timelines for them all. So we didn't wait for the mice to get super sick or where the tumors were massive, but we tended to look around 8 to 10 days, depending on the model, where there was clearly disease, but it wasn't extremely advanced. It was still relatively early days. And what we found was that regardless of the disease, if we looked in the synovial fluid for the arthritis mice, in the tumor itself for the tumor-bearing mice, or in the skin of the ear, we could find cells that came from the colon or that were in the colon 24 hours before. And this is interesting because if you remember, the skin was actually one of the tissues where we had not really found migratory cells. But if there is disease in the skin, we can now find migratory cells in the skin, which this would initially tell us that these cells are sort of amenable to changing and responding to different stimuli. And as a final piece of data, when we started looking at the identity of those cells across those various sick tissues, is that the identity of the cells was quite different. So if we look, let's say, in the synovial fluid, each bar here represents a mouse, and then the different immune cells are color-coded. So you can see here that in the synovial fluid, the majority of immune cells were B cells. But if we look in the tumor, then suddenly what we're seeing is a lot more disease, macrophage, gamma delta T cells, more myeloid-like populations in the tumor. And then if we look in the skin of the ear, there's a lot more CD4 T cells and gamma delta T cells, barely any B cells we could detect in the skin, which is extremely rare. And whereas in the spleen, we find, profiles that we would expect regardless of the disease state. So, with that, we feel that it's just the tip of the iceberg. So, of course, there's a lot that we don't know, but what we hope to have demonstrated at this point is that there really is a sort of massive efflux of cells from the gut that have the ability to reach other tissues, that have the ability to retain some of the phenotype that they've acquired in the gut, and that they have the ability to respond to insult, like for instance, as we see in the skin. We still don't know exactly how any of this is regulated or what exactly is the function of these cells and what it is that they're doing exactly. But I do think that, we are hopefully going to be moving away from these various specific axes to more of a generalized phenotype of these cells across tissues. And with that, like I said, I think this is a very exciting area, which of course I'm biased, but there's a lot of room for discovery here. And I just opened up my lab this week, and we are recruiting. We are in Worcester, Massachusetts. So you have access to Boston and New York, but it's a lot cheaper. So if you're looking for opportunities and you're interested in this area, feel free to shoot me an email. And with that, I'd just like to finish by thanking my postdoctoral mentors, Christoph and Diane. I loved my time in the CBDM lab, and I learned a lot from them. So I'm very thankful for them to have given me the opportunity and to have enabled me to make it to the point where I can open my own lab. And then a lot of thanks to Angela Magnuson, who initiated the KIDA project protocols, like I said, say about a decade ago. Our lab manager, Adriana, who keeps the lab running, Brenda for bioinformatic help. And there are other people in the lab that also do a lot of gut group, a lot of gut immunology work, and we work closely together. Just Shika, who's now an independent PI at the SOC, Martina, who's now in a director, I think, company, Yang Shinshin Adelaide, and also my baymate, David Owens. And I'd like to thank EMBO for funding as well. And with that, I'd be happy to take any questions.
Fantastic work, Silvia. So we have actually some questions already. So Aitak Farsi from chat, she asked, very interesting. Did you also measure migration to brain, borders, meninges in any of your models?
So we tried measuring migration to the meninges, but what happens is the KIDA is great. It's a great model, but it also has its limitations. And one of the limitations is that we cannot photoconvert the entirety of the gut, right? We can only photoconvert a portion, not 100%. The efficiency is 50 to 60% photoconversion of a portion of the gut at one time. And then from that, you have to extrapolate that not all those cells are going to migrate. So on a good day in the spleen, you're hoping for 0.5 to 1% of the cells are going to come from the gut. So whenever we looked in the meninges, we didn't have thousands and thousands of cells to work with. So I think that the threshold to be able to actually detect if any of those were coming from the gut just wasn't there. We saw some hints of, oh, maybe there's one red cell, two red cells, but we didn't really have the confidence at the time to be able to say yes, or there's cells migrating or no. But we never looked in the whole brain. We only did meninges.
Taking that going through that question, did you do EAE experiments?
No. So again, to add to that, we only looked in the meninges on a healthy 6 to 8 week old mouse. So yeah, I don't know. Actually, we did EAE. We did one random EAE experiment, but we didn't actually look in the brain. We looked at spleen and lymph nodes, and it seemed like there was a lot happening, but then postdoc, it was a collaboration with another lab, it got dropped. So I don't really have EAE data.
And so I have two questions regarding what is the long-term phenotype of these T cells that migrate from the gut? Do you expect that they will recirculate or they will stay there? And if so, perhaps they were, because we always think at the gut, that place where it's allergenic. And I was thinking about the EAE because there was a paper from Jennifer Gommerman, who did EAE as well. And she said like there were B cells, IgA, IL-10 producer. So I was thinking that perhaps all of these phenotypes you see, perhaps these cells are become allergenic, and also the paper from your lab from the muscle tissue where, you know, the tissue repair. What is your thought about that? What is the phenotype of these cells? Are they staying there? Or they're circulating? What is your thought?
So this is just thinking. I don't have so far any data to support it, but to me, I always like to interpret it as, you know, we learned about immune cells developing in the lymph nodes and in the thymus, T cells get educated in the thymus, B cells get educated in the bone marrow, and then they leave and they circulate. But a lot of that huge amount of knowledge predates the microbiome. So to me, it's not, so a lot of it is antigen specific from the context of, oh, there's this huge infection, right? So it makes sense. But I feel like a lot of how we built our immune cell knowledge didn't factor in the fact that we have a lot of antigens in our gut, right? So to me, it's not far-fetched to think, yes, the cells developed in the thymus, they develop in the bone marrow, but maybe they didn't have, that's primary school. Then they go to high school in the gut, where they get primed and develop these other more tolerogenic phenotypes that, because we know that things escape the gut, right? We know that there's metabolites and proteins that reach other places in the body. So to me, it's not far-fetched to think that, yes, the gut, maybe it's a site for building tolerogenic responses that can then be used by other tissues. But I have, that's just the thought.
Thank you so much. I'm very impressed about this work. So there's another question from Christina. She asked, what is the phenotype of the T cells that are migrating from the gut? Are these TRM, or there are transient gut cells that exist there? Also, she asked if there's any T helper subtype preference to migrate from the gut. Perhaps they are Th17 cells or Tregs migrating from the gut to the other tissues. Or any particular markers that you can identify?
Yeah, so there's sort of two branches of the T cells that we clearly see that are migrating out. One is the sort of more IEL type phenotype. And of that, I have very little else information. It's quite obscure. We are starting to see evidence, not me, but a colleague, of perhaps it's not that rare. Perhaps we are starting to see IELs in other tissues unexpectedly. Excuse me. And there's not much I can say about that. Now, on the other hand, we do see this cluster of RR gamma T cells. And we do have, like you say, another paper from the lab where we found RR gamma T cells responding to injury in the muscle. If you look at the literature of cells migrating out of the gut, a lot of it is Th17 related in terms of disease. Now, we actually didn't find Th17 cells in the steady state mice when we did the profiling, but we do find IL-17 producing gamma delta T cells. So I actually think, and I wonder if a lot of the RR gamma related phenotypes, whether it's RR gamma Tregs, whether it's IL-17 producing gamma deltas, Th17s, are basically just a gut derived immune cell population. And so in that case, then yeah, I do think that they recirculate. I do think that perhaps they go through the gut, they need certain survival signals that they see there, and then they go. If they find something else to do, they go do it. Otherwise, they perhaps return to the gut. But this is something that we wanted to study, but with the Caeta, it doesn't have the resolution to really detect if a cell has come back to the gut. So I'm still figuring out how exactly we can do finer tracing and figure that out. But it's definitely interesting to think about.
So there's another question. So in the chat from Gata Rubina, and she said, do you think that differential abundance of KATE positive gut immigrant cells in peripheral tissues can be impacted by different half-lives of KATE in each cell population? Like, for example, intrinsic degradation or divisional dilution?
For sure. I think that it's sensitive enough that's why a lot of the times we look at 24 hours, because there's not much that can happen. I mean, maybe not innate immune cell in terms of signaling, but there's so much caeta in these cells that there's not much that can happen within 24 hours that will cause it to, you know, disappear. But it is entirely possible that there's more, you know, obviously an activated cell, a cell that's differentiating, it's going to have a lot more protein turnover than one that's a bit more early on in its transition. And so it's entirely possible that our study so far has been biased to pick up the sort of less differentiating, less effector type cells. I don't think we missed them completely. Like the single cell showed, you know, we can pick up activated effector cell phenotypes, but in terms of numbers, it is perhaps biased to more stagnant populations. So yeah, I think that we're not going to miss them in that 24-hour window, but it's possible that our ratios and percentage-wise are going to be biased for certain cell types. Yeah, that's possible.
So Silvia, I have a final question. So when you challenge the animal, you find in the skins some cells from the gut. So there's another tissue that at the homeostatic conditions you don't even found any T cells, which is the thymus. And I'm thinking about the Gretchen dial work where she found that there are some migration from dendritic cells to the gut to promote clonal selection of T cells. Did you find any sort of these things in a challenge animal?
No, we have not looked at that. We did discuss it, and it's entirely possible. I don't think that's why I'm resistant to say, oh, there's no migration to the thymus. I don't think we can say that. I think that in our experimental conditions where I'm not running the entirety of the thymus, I'm not enriching for disease, I'm not, you know, I'm trying to sort of equilibrate across tissues. If there is migration to the thymus, it's negligible compared to the others. But it doesn't mean that's not there. As we've learned from T-Rex, numbers aren't always the, you know, the win it all. T-Rex are a tiny population in a lot of instances, but they make a huge difference. So, you know, that could easily be the case in the thymus. And like you said, we didn't actually look in perturbations in the thymus. So it's possible that in that case, like it is for the skin, that we'd actually see a lot more communication between the two.
So thank you very much, Silvia. I think there's no more question in the chat. I want to say congratulations and everyone that joined today. So in the chat, I put some, if you want to join our Slack group or Facebook or Twitter, there are a lot of seminars that we are promoting there. And the best of luck, Silvia, in your future endeavors. And I really will follow your work closely. Thank you so much. And thank you, everyone. Bye-bye.